All right, so today we are going deep on something that I think is really, really cool.
Oh, yeah.
And that is how certain antibiotics target and actually just break apart bacterial membranes
Yeah.
in our sources, go deep on this, on a molecular level. You know what I mean?
Oh, yeah, absolutely.
It's some pretty fascinating stuff.
It's really cool how these things work.
Yeah. So just to remind everyone, bacterial membranes aren't just there, right? They're super important for the bacteria to do everything it needs to do to stay alive.
Yeah, they control what goes in and out of the cell.
Exactly.
They're involved in all sorts of metabolic processes.
Yep. And so if you can disrupt that,
The cell just dies.
the cell's toast.
Rapid cell death.
Yeah. And so what we're going to look at today are two main classes of antibiotics that actually do this.
Right.
Polymyxins, you might have heard of colistin, that's one. And then daptomycin.
Yeah.
And we're going to figure out how they work, how they do this magic on bacterial membranes.
Yeah.
Or why sometimes it's risky, even though it's effective.
Yeah, there's always that trade-off with the toxicity.
Yeah. Every drug has a side effect.
Right, right.
Exactly.
So let's unpack this.
Yeah, let's get into it.
Let's start with polymyxins.
Okay.
So these mainly go after gram-negative bacteria, right?
Right.
And so what makes them so special?
Well, gram-negatives,
Yeah.
you've got that extra outer membrane.
Yeah, that outer membrane.
The LPS.
The LPS, it's like their armor, right?
Yeah, lipopolysaccharide, the LPS, that's right.
They're
Tough!
They're tough little bugs.
And that LPS is actually what these polymyxins are going after.
Right.
So, okay, picture this.
Right.
Polymyxins are cyclic lipopeptides.
Okay.
So it's like a circular chain, and then it's got this fatty acid tail, and they have a positive charge.
Okay. So,
And this is where it gets so cool, right?
right. This is the cool part.
Because the LPS is negatively charged.
Right.
So they are attracted to each other, like magnets.
Oh. They're just drawn right to it.
So they bind to the outer membrane of the bacteria
Yeah.
because of those charges.
Exactly.
Opposite charges attract.
It's like basic chemistry in action, right?
Exactly.
But here's the thing.
Yeah.
It's not just that they bind.
No.
It's what happens after.
Right.
So these positively charged polymyxins, they actually displace other ions.
Yeah, like magnesium and calcium ions,
Exactly.
which normally stabilize the LPS layer.
So they're like the glue. These magnesium and calcium ions are the glue.
Yeah, think of them like rivets holding everything together.
That's a good analogy.
And the polymyxins come in and knock those rivets out.
Yeah, and so without the glue, without the rivets.
The outer membrane becomes weaker.
And so what
It
happens?
destabilizes.
It destabilizes.
It basically starts to fall apart and
Yeah.
it becomes much more permeable.
So for you listening, imagine like cracks forming in that armor.
Exactly.
And now there are these weak points.
And that's where that fatty acid tail comes in. Oh, the fatty tail, right?
Right. It
Oh,
sneaks in into the cytoplasmic membrane, the inner membrane.
Right. So there's the outer membrane and the inner membrane,
Yeah.
and it's disrupting both.
It's like a wedge, just forcing its way in.
Oh, I see.
Yeah.
And so what happens to the bacteria then?
Well, basically it all falls apart.
Like literally its insides start leaking out?
Yeah. Leakage of intracellular contents.
Wow.
They lose their membrane potential.
And membrane potential, that's like their battery, right?
Yeah. It's the electrical gradient across the membrane. It's essential for all sorts of cellular functions.
Oh, so without that,
The cell can't function.
it dies.
It dies rapidly.
Yeah, it's like lights out.
Exactly.
And our sources call this a bactericidal effect.
That's right. It kills the bacteria.
So it doesn't just stop it from growing.
No, it actually kills it.
It's like a seek and destroy mission.
Yeah, pretty effective.
Pretty effective.
But they do have a limited range.
Yeah, so tell us about that.
So they mainly work against those multi-drug resistant gram-negative bacteria.
The tough ones.
The really tough ones, yeah, like Pseudomonas, Acinetobacter, Klebsiella.
Those are some big names.
Yeah, those are some nasty bugs.
But they don't work against everything, right?
Right. Not against gram-positive.
Those don't have that outer LPS layer.
Exactly. That's their Achilles heel.
And even some gram-negatives are resistant.
Yeah, like Podius and Serratia.
Huh, interesting.
Yeah.
But, okay, so this sounds pretty great, right?
Yeah, it's a powerful tool.
Powerful tool.
But there's a but.
There's always a but with
Right.
drugs, so what's the downside here?
Well, they can be toxic to us, too.
Uh-oh.
Yeah, especially to the kidneys and the nerves.
So they can cause kidney damage and nerve damage.
That's why colistin is often a last resort.
Yeah, you don't want to use it unless you really have to.
Right. Only when the other options have failed.
Got it. Okay.
Right.
So that's polymyxins.
That's polymyxins.
Let's move on to daptomycin.
Okay.
So this one, it goes after gram-positive bacteria.
Right. It targets their cytoplasmic membrane.
So the inner membrane.
Yeah.
So it's not going after that LPS layer.
No, because gram-positives don't have that outer membrane.
Right. Right.
So daptomycin has to use a different strategy.
So what's its strategy?
Well, it's also a cyclic lipopeptide.
Okay. So similar structure to polymyxins.
Yeah, but its mechanism is different.
Okay. Tell me about it.
So it needs calcium ions to work.
Calcium ions. That's interesting.
Yeah, it's crucial for its action.
So what do those calcium ions do?
They help daptomycin bind to the membrane and insert its lipid tail.
Oh, so calcium is like the key that unlocks the door.
Yeah, think of it as the trigger.
The trigger.
That allows daptomycin to anchor itself.
Okay. And once it's anchored, what happens then?
Well, our sources mention oligomerization.
Oligomerization, what's that?
It means that multiple daptomycin molecules cluster together.
Oh, so they team up.
Yeah, they form a team.
Like a little gang of daptomycin.
And this team creates a disturbance in the membrane.
A disturbance, okay.
Yeah, they might even form channels.
Channels.
Yeah, basically holes in the membrane.
So stuff can leak out.
Yeah, but in this case, it's mainly potassium ions that flow out.
Potassium ion, so it's not just a random leak.
No, it's the specific ion that's affected.
Huh. And why is that important?
Well, it disrupts the electrical gradient across the membrane.
That membrane potential again?
Yep, it depolarizes the membrane.
It messes with the battery.
Exactly.
What does that do to the bacteria?
Well, it has a domino effect.
A domino effect, right?
It inhibits the synthesis of essential molecules.
Like what kind of molecules?
DNA, RNA, proteins, all the things a cell needs to survive.
So it can't make the building blocks of life anymore.
Right, and without those, it dies.
So it's another bactericidal effect.
Yep, but there's a difference here.
Oh, what's that?
With daptomycin, the bacteria doesn't lyse.
The lyse, you mean burst open?
Yeah, it just sort of shuts down.
Huh, that's interesting. So, okay, daptomycin is another powerful antibiotic.
It is.
But does it have a narrow spectrum too?
Yeah, it only works against gram-positives.
Okay, so it's like the opposite of polymyxins.
Right, they each have their own niche.
And does it work against the really tough gram-positives?
Oh yeah, it can kill MRSA and VRE.
Wow, those are some serious superbugs.
Yeah, they're resistant to a lot of other antibiotics.
So daptomycin is like the big gun?
You could say that.
But are there any downsides?
Well, it can cause muscle toxicity.
Uh-oh, that doesn't sound good.
Yeah, it can cause muscle pain and weakness, even muscle breakdown.
Yikes.
So doctors have to monitor patients carefully.
Yeah, check their blood for signs of muscle damage.
Right, exactly.
And is there anything else we need to know about daptomycin?
Yeah, one more thing.
Okay.
It's inactivated by pulmonary surfactant.
What does that mean?
It means it doesn't work in the lungs.
If you can't use it for pneumonia.
That's right.
Interesting.
Yeah.
Okay, so we've got these two very different antibiotics. Polymyxins going after the outer membrane of gram-negatives.
Right.
And daptomycin targeting the inner membrane of gram-positives. Different strategies, but the same goal.
Disrupt the membrane, kill the
Right.
bacteria. It's a
God.
clever approach.
It is, but it makes you wonder,
What's that?
why are these drugs sometimes toxic to us?
Well, it all comes down to the fact that bacterial membranes
Yeah.
and our own cell membranes,
Mm-hmm.
they're pretty similar.
Oh, that makes sense.
Yeah, the basic structure is the same.
So if a drug can disrupt a bacterial membrane,
It can potentially disrupt our own cells, too.
right, so there's that risk of collateral damage.
Yeah, that's the trade-off.
And so it's a huge challenge for scientists
It is.
to develop drugs that are super specific of bacterial membranes.
And don't harm our own cells.
And that's really where the future of antibiotic research is headed, right?
Absolutely. That's the holy grail.
It's so cool to think about, like, what other ways are there to
Yeah.
target these membranes?
Yeah. What other clever strategies?
Right, like what hasn't even been discovered yet.
Who knows what the future holds?
Yeah, it's exciting.
It is.
It's an exciting field to be in.
Absolutely.
So for all of you listening, I want you to think about this, okay?
Right.
Cell membranes are like the fundamental building blocks of life, right?
Yeah. All living things have them.
So what other clever ways can we target bacterial membranes and at the same time keep ourselves safe?
Yeah. Minimizing the harm to the host.
It's a challenge, but it's a challenge worth tackling.
Absolutely.
Definitely food for thought.
Food for thought.
All right, so if you want to dig deeper into this, check out these tags, okay?
Okay.
#PharmaBio, #antimicrobial, #mechanism of action.
Yeah. Lots of good stuff out there.
Yeah, thanks for joining us for this deep dive.
Always a pleasure.
All right, we'll see you next time.
See you later.
Bye.